Hi everyone, in this video, we will learn regulation of gene expression in bacteriophage lambda. What do you mean by gene expression first of all? Gene expression is nothing but transcription and translation. So we will see how bacteriophages, particularly bacteriophage lambda, regulates its gene expression, regulates its transcription and translation. So I am recording this lecture now. Once my recording is over, I'll switch off the laptop. I will switch off the lights and fans because I'm not using them. I'm not sitting in the room after that to save energy. Similarly, bacteria, bacteriophages, they regulate their gene expression. So let's talk more about lambda phage. Let's see the early transcription events. Now, what is the genetic material of lambda phage? The genetic material of lambda phage is linear double-stranded DNA with 12 nucleotide long sticky ends. So inside the phage, the genome is linear double-stranded DNA with 12 nucleotide long sticky ends which are complementary to each other. Once the lambda phage injects its DNA into the host E. coli, once in the host, this linear DNA can circularize by pairing of the sticky ends. You can see the cos sequence, and the nicks are sealed by host DNA ligase. Now, lambda phage is a temperate phage. What do you mean by a temperate phage? It can undergo either a lytic cycle or a lysogenic cycle. It has a choice. So when the phage infects a bacterial cell, it has a choice of whether to enter the lytic pathway or lysogenic pathway. In the lytic pathway, the phage behaves as a virulent one, and it will do the replication, carry out lots of protein synthesis. Progeny phages will be assembled and they will kill the cell, host cell, and will come out to infect other host cells. Whereas in lysogeny, the genome of the phage is integrated into the host chromosome and it replicates along with that, and the phage is said to exist in a prophage state. So we will study this genetic switch where the phage will decide whether it has to enter the lytic pathway or the lysogenic pathway. This is the genetic map of lambda phage, and initially only two promoters are active, promoter left and promoter right. So why this promoter left and promoter right are called as PL and PR? This PL stands for promoter left and this PR stands for promoter right. So transcription begins at promoters PL and PR by the host RNA polymerase enzyme. PL promoter left is for the leftward transcription of the left early operon, and promoter PR is for the rightward transcription of the right early operon. So from this PL, I'll use a different color for PL. From this PL, the RNA polymerase will sit at PL and will move in the leftward direction. And the first gene to be transcribed is the N gene. And from PR, the RNA polymerase will move in the rightward direction. And the first gene to be transcribed is CRO. So the first gene to be transcribed from promoter right is CRO, the product of which is the CRO protein. And this protein plays an important role in setting the genetic switch to the lytic pathway. So if CRO dominates, lytic pathways follow. The first gene to be transcribed from PL is N. N protein is a transcription anti-terminator. When I say anti-terminator, it doesn't allow the termination. So it allows things to continue. It's an anti-terminator, and it's anti-terminator of transcription. So it doesn't allow the transcription to be terminated. It allows transcription to continue. It allows mRNA synthesis to proceed past certain transcription terminators. And there are 4 genes that are transcribed due to the action of N. And those 4 genes are C2, O, P, and Q. Out of these 4 genes, 3 of them are involved in the lytic cycle, which is O, P, and Q. And 1 of them is involved in the lysogenic cycle, that is C2. C2 encodes protein C2. The C2 protein will turn on the transcription of 2 more genes, which is C1 repressor and INT integrase. And this C1 repressor and INT are involved in lysogeny, and that's why we say that C2 is involved in lysogeny. Whereas O, P, and Q are involved in the lytic cycle. O and P encode replication proteins, and Q is another anti-terminator protein needed to turn on the late genes for lysis and phage particle proteins. Now, these are the early transcription events with the help of another diagram. So once the lambda phage has injected its DNA, the linear DNA is circularized, but here we are showing a part of that circular chromosome. And the phage growth begins when RNA polymerase binds to early promoters PL and PR, making N and CRO genes. Throughout this figure, the inactive promoters are shown by a pink color, and the active promoters are shown with the help of yellow color. So you can see the lambda phage has many promoters, PI, PRM, PRE, PR prime, but all of those promoters are, you can see pink in color, they are inactive. Initially, only 2 promoters are active, and they are the promoter left, which you can see in yellow color, and promoter right, which you can again see in yellow color. So initially only these 2 promoters are active, promoter left and promoter right, and the RNA polymerase can sit on these 2 promoters. So if the RNA polymerase sits at promoter left, which is this one, the RNA polymerase sits at promoter left, which is this one, it will move in the leftward direction, and the first gene to be transcribed will be the N. And if the RNA polymerase sits at promoter right, which is this one, it will move in the rightward direction, and the first gene to be transcribed from promoter right is CRO. CRO is involved in lytic cycle, whereas N is the transcription anti-terminator. It allows transcription to continue, resulting in the transcription of 4 more genes, C2, O, P, and Q. C2 ensures that C1 and INT are transcribed. So those C2, C1, INT are involved in lysogenic cycle, whereas O, P, Q lytic. After that, 2 competing pathways may occur. Either the phage may go for lysogeny, which is integration of the phage chromosome into the host chromosome, or the phage may go for the lytic cycle, where it will make progeny phages and kill the cell. Now, what decides whether lysogenic pathway will be followed or lytic pathway will be followed? If C1 repressor dominates, then lysogeny will be established, whereas if CRO protein dominates, then lytic pathway will be established. Let's see how the lysogenic pathway is established. The establishment of lysogeny requires the products of C2 and C3 genes. So C2 protein stabilized by C3, and the C2 protein, which itself is made because of action of the N protein. We have told you that initially only 2 promoters are active, promoter left and promoter right, and the RNA polymerase will sit at promoter left, move in the leftward direction, and the first gene transcribed is N. The N protein is a transcription anti-terminator, and it will allow the transcription of 4 more genes, and one of them is C2. And C2, which is made because of action of N, will activate 2 promoters here. C2 will bind to promoter PRE, promoter for repressor establishment, and also for promoter I. Now this promoter I, PI, and promoter RE, you can see now they are yellow in color. That means they are active. But if I take you back to the initial step when the phage infects E. coli, initially when the phage infects and the chromosome is circularized, at that point, you can see that the promoter PRE is pink in color and promoter PI is also pink in color. That means this PRE and PI are inactive initially. So are the other promoters. The only two promoters active in the beginning are PL and PR, that is promoter left and promoter right. But now, if the lysogeny is established, this promoter PRE and PI are being activated, and they are activated by none other than C2. So C2 is binding and activating promoter PRE and PI, and RNA polymerase will sit at promoter PRE, will move in this direction to ensure the transcription of C1 gene, which is the repressor. Now, you would say that the PRE promoter, if RNA polymerase will sit at PRE and will move in this direction, C1 to baad mein aayega. Immediately next to PRE in the left is CRO gene, and CRO is for lytic cycle, and we are talking about lysogeny. But CRO will not be transcribed here because, as I told you, CRO is made in another direction, which is this rightward direction from a different promoter right. So CRO will not be made here. C1 will be made, and from promoter PI integrase would be made. So C2 protein stimulates the synthesis of mRNA from PRE for C1 gene and from PI for integrase. Once C1 and integrase are made, what next? This C1 repressor binds to operator region. Which operators? C1 is going to bind to operator left, OL, and operator right, OR. Now, if I take you back to the genetic map, where are operator left and operator right? Operator left is in close proximity with promoter left, whereas operator right is close proximity with promoter right. And this operator left is so close to promoter left that their sequences overlap. Similarly, the operator right is so close to promoter right, their sequences overlap. So if someone sits at operator left, there is no place for anyone to sit on promoter left. Someone sits at operator right, there's no place for anyone to sit at promoter right. So if I have a chair at home, I sit on that chair and I occupy so that no one else can sit. So if I am the C1 repressor and the chair is the operator promoter sequence, I am being C1 repressor, will sit on the chair, that is the operator promoter, so that someone else cannot occupy that chair, that someone else is RNA polymerase. So since the C1 repressor is sitting at the operator left, RNA polymerase cannot sit at promoter left. And similarly, since C1 repressor is sitting at operator right, RNA polymerase cannot sit at promoter right. And can I say that promoter left and promoter right are inactivated? Okay. So we are into this slide. Operator left and operator right. So C1 repressor binds at operator left and C1 repressor binds at operator right. And since RNA polymerase cannot sit at promoter left and promoter right, promoter left and promoter right are inactivated now. So initially the two promoters which were active, promoter left and promoter right, they are being inactivated in lysogeny. And which genes used to be transcribed from these two promoters? From promoter left, the first gene to be transcribed was N. But since promoter left is inactivated, N will not be made. And N used to result in the transcription of 4 more genes, O, P, and Q. They will not be made because N only is not there. So that's good because lysogeny is being established and O, P, Q are lytic proteins. C2 will also not be made, which was a lysogeny protein, but because N is not there for the simple reason, but C2 has already done its job. It has activated promoter RE and promoter I, and C1 is made. So C2 is not needed now. And since promoter right is also inactivated, the first gene to be transcribed from promoter right was CRO, which was the major lytic cycle protein, so it is not made, and lysogeny is established. So that, and also not only this repressor by binding to operator right and operator left, inactivating the promoter left and promoter right, this repressor C1 will activate one more promoter, that is promoter PRM. And initially, if I take you to the first slide, promoter PRM is inactive initially, it's pink in color. But now the promoter PRM for repressor maintenance, it is activated, yellow in color, by none other than repressor itself. Okay, and from this promoter PRM, the C1 repressor would be made. So abhi tak C1 repressor promoter PRE se ban raha tha. But now the C1 repressor would be made by from promoter PRM, and this remains the only active promoter in lysogeny. All the promoters are inactivated. PRM remains the only active promoter. The sufficient repressor binds to OR and OL, blocking the RNA synthesis, and repressor bound to OR stimulates the synthesis of more repressor from PRM, integrates, promotes the integration of the phage DNA in the chromosome DNA, and lysogeny is established. So summary, all these proteins are being made in E. coli, but who wins the race? It's like that. If CRO is synthesized first, or if CRO dominates, we know lytic pathway will be followed. But at the same time, N is also made, its transcription anti-terminator. It results in the synthesis of C2O, P, and Q. Some are lytic proteins, some are involved in lysogeny. But if C2 wins the race and activates PRE and PI, and C1 repressor is made, C1 repressor will bind to operators left and right, inactivating promoter left and right. CRO and N will not be made, and lysogeny will be established by integration of the phage DNA into chromosomal DNA by integrase. So this was how lysogeny is established. Let's see how a lytic pathway is induced. There are certain inducers such as ultraviolet light which damage the DNA. We know that. But this ultraviolet light damage changes the function of a multifunctional bacterial protein called REC-A. REC-A normally is involved in recombination, but REC-A, because of UV damage, its function is changed, and it will stimulate the lambda repressor. Which is lambda repressor, or what is lambda repressor? The C1. C1 is the lambda repressor which is bound to operator left and operator right. REC-A will stimulate lambda repressor to cleave itself, और वो वहां से हट जाएगा operator left or operator right. Now the resulting absence of the C1 repressor from the operator left and operator right, because it has cleaved itself, can I say that if I was the C1 repressor and I was sitting on the chair, but I got up now, so the chair is free. Similarly, C1 cleaved itself and the operator left and operator right are free. So is promoter left and promoter right. So RNA polymerase can bind to promoter right and the CRO gene can be transcribed. The CRO protein made will decrease the synthesis from PL and PR. So वो PL से इसके लिए decrease करेगा synthesis so that C2 is made less. This reduces the synthesis of C2 because you know from PL N is made and N results in the transcription of C2. So this is to reduce the synthesis of C2, which is the regulator of C1 lambda repressor, and the CRO protein directly blocks the synthesis of C1 repressor from PRM. At the same time, the transcription from the right early operon from PR is decreased, but enough Q proteins are accumulated to set the genetic switch to starting the lytic pathway. So this operator left, here C1 was occupied, operator right, यहाँ पे भी C1 was occupied or sitting, but since because of UV light and change in the function of REC-A, C1 cleaved itself, C1 is not here now, operator left, operator right are free. So are promoter left and promoter right. So you can see PL and PR again yellow in color. CRO will bind to PL and PR. CRO will ensure, CRO only will bind to PL and PR, and CRO will, by occupying operator OR and OL. Now CRO will sit at operator left and operator right. It will reduce the synthesis of C2, and it will also prevent the synthesis of this repressor from PRM. You can see PRM is inactivated, and this allows enough Q proteins to accumulate, and Q protein activate promoter PR prime. This is not PR, this is PR prime. So Q activate promoter PR prime, stimulates the late gene transcription required for the synthesis of structural phage proteins and lysis proteins, and the lytic cycle will be followed. What is summary? Lambda uses complex regulatory systems to choose either the lytic or lysogenic pathway. It has a choice, it has a genetic switch. And this decision depends on a sophisticated genetic switch that involves competition between T1 repressor and the CRO protein. If the repressor dominates, that is the C1, you know what would happen? It would occupy the operator left and operator right. It will inactivate promoter left and promoter right. It will keep only one promoter active, that is PRM, followed by integration of the phage DNA, that is lysogenic pathway will be followed if the repressor dominates, and it doesn't allow CRO protein to be made. If repressor dominates, it doesn't allow the CRO protein to be made because CRO is involved in lytic cycle, and repressor dominates, lysogeny is followed, and also the PR is inactivated. On the other hand, if CRO dominates, then the lytic pathway will be followed, and it will not allow the repressor to be made. So this is the regulation of gene expression in bacteriophage lambda.